Good evening to all. Welcome to the international webinar entitled COVID-19 Pandemic Needs, Science and Hope, organized by the Department of Botany in collaboration with IQAC. Warm welcome to all. Today among us, we have the chief motivator of this webinar, our officer in charge, Dr. Shobhan Yogi, and IQAC coordinator, Dr. Amoresh Mandor, the chief advisor of this webinar, Dr. Kalidas Raj, and two other distinguished speakers, Dr. Manishankar Bhattacharya, senior scientist of Institute of Microbial Technology, and Dr. Devojyoti Ghosh, research scientist, University of Cincinnati College of Medicine. Once again, I welcome all to this webinar. May I please request Dr. Shutapa Sanyal, convener of this webinar, to give an introduction. Over to Shutapa. Dr. Shutapa.
সুতপাদি মিউট আছে।
Am I audible?
Yes. Yes.
শোনা যাচ্ছে?
Yes.
Yes. Yes.
Okay. Okay, sir. Thank you. Good afternoon, everyone. Good afternoon, everyone. COVID-19 pandemic is increasing day by day, and its death toll is increasing day by day. We are getting horrified by receiving different information from different sources. This information actually makes us mentally ill. The basic thing is that our researchers has not sparing any time or any resources to defeat this new coronavirus. We must know all the rays of hope associated with COVID-19. Not the big, but the science associated with COVID-19. Botany Department of Krishnagar Government College, in collaboration with IQAC of Krishnagar Government College, has organized an international webinar to give us a scientific idea about the COVID-19. I, Dr. Shutapa Sanyal, Department of Zoology, Krishnagar Government College, welcome our two distinguished speakers for today's webinar. Dr. Manishankar Bhattacharya, Senior Scientist, CSIR Chandigarh, and Dr. Devojyoti Ghosh, Research Scientist, Cincinnati University, USA. Now I welcome our officer in charge and request him kindly to inaugurate officially our webinar so that it's delivering his welcome address. Thank you once again for joining us, and over to our vice chair.
Thank you, Dr. Namaskar and good morning, everyone. Who are present in this platform. Is it audible? শুনতে পাওয়া যাচ্ছে? Hello, hello, hello.
Yes.
আমি repeat করছি. Namaskar and good evening to you all present in this web platform. Today 1 in Department of Botany and IQAC of our Krishnagar Government College. I wholeheartedly welcome our two guests with CSIR Institute of Microbial Technology, Chandigarh, India, and Dr. Debojyoti Ghosh, Fellow with the American Academy of Allergy, Asthma, Immunology with Cincinnati University, USA. And especially good morning to Dr. Ghosh because it's early morning in US. I also welcome my beloved students, my colleagues from my college, and all other students and teachers who have joined this international webinar from different colleges and institutes. Our college is going through a process which is due to the its 175th year of establishment. We started the celebration of 175th year of establishment last year in November 2019. So we are within the year of 175th year, though we had a plan to celebrate this great occasion with so many programs, but due to this restrained ourselves because all the institutes, colleges are closed. Only this option of webinar is now with us. I'm really glad I've already talked for us in our webinars. Today's topic is the COVID-19 pandemic, myths, science, and hope. So research persons who are associated with this biological system, definitely all the viewers will be benefited to a great extent to understand this COVID problem, why it is so, and how we can overcome. Once again, I welcome Dr. Monishankar Bhattacharya,
Institute. He joined as a scientist in the same institute in 2011. He did his undergraduate and postgraduate studies in Vishwabharati and submitted his thesis in National Institute of Pharmaceutical Education and Research, Mohali, in the Department of Pharmaceutical Technology and Biotechnology. He has been awarded with several national scholarships and fellowships during his graduate and postgraduate studies from the Government of India. He remained as a MIUR Fellow from 2006 to 2009 from Italian Ministry of University and Research. He also had a training in advanced course in downstream processing from Technological University Delft, Netherlands in 2007 as an assistant professor of Academy of Scientific and Innovative Research, Chandigarh, and IMTech JNU Joint PhD program. He also teaches basic organic chemistry, bioinstrumentation and biotechniques, bioorganic chemistry, and biochemical engineering. His research interests are fermentation of surface tension lowering and micelles forming compounds and downstream processing, biocatalysis, immobilization and stabilization, development of novel antifungal compounds, development of antifungal and antibiofilm products, application of nanostructured materials in biocatalysis and drug delivery. Two of his have obtained their PhD degrees under his supervision. Presently, he is guiding 8 students as guide or as a co-guide. Manishankar is actively engaged in school student outreach program through Jigyasa, providing training to the masters and graduate students through internship program. I welcome you, sir. It is a great honor that you have accepted and given your consent in spite of your busy schedule. Over to Dr. Manishankar Bhattacharya. Hasn't joined yet. So can we continue with
Shormishta, did you call him? Maybe some technical issue.
Okay, can we continue with Dr. Devojyoti Ghosh? Has he joined?
Initially, he
Yeah.
joined.
Yes, he's here.
Huh?
Yes, Dr. Ghosh is here.
Yeah, Dr. Ghosh has joined. Should we continue with him?
Can you talk with Dr. Bhattacharya?
Yeah, I'll be talking with him now. Yeah.
Hello, I can present now.
Has he joined?
Not yet.
Has Manishankar joined?
কৌশি মণিশঙ্কর বাবুর কাছ থেকে জয়েনিং রিকোয়েস্ট এসেছিল গুগল মিটে? আচ্ছা, কোনোভাবে টেকনিক্যালি কিছু
না, ও জয়েন করতে পারেনি।
তাহলে ডক্টর ঘোষ প্রথমে স্টার্ট করে দিন, ওনাকে তুমি বলে দাও যে আমি এটা শেষ হলে সেকেন্ডে করুন, তাই না?
কৌশিক, হ্যালো কৌশিক, হ্যাঁ কৌশিক, তুমি ওর যে ওর জিমেল অ্যাকাউন্টটা আছে তোমার কাছে? এই মনির জিমেল অ্যাকাউন্টটা আছে তো? জিমেল অ্যাকাউন্টে একটা ফ্রেশ তোমার ইয়ে পাঠিয়ে দাও, তুমি ওই লিংক পাঠিয়ে দাও ওকে। হ্যাঁ হ্যাঁ ঠিক আছে। আচ্ছা ঠিক আছে।
ওই @ac.in এটা কিন্তু গুগল মিটে ভ্যালিড।
So let us continue with Dr. Ghosh. Sir,
Yes.
our next
Yes.
speaker is, as Dr. Monishankar Bhattacharya hasn't joined yet. Very sorry, he'll be joining later. Our next speaker is Dr. Devojyoti Ghosh, research scientist of University of Cincinnati College of Medicine. It's my great pleasure to introduce Dr. Devojyoti Ghosh, fellow of the American Academy of Allergy, Asthma, Immunology, and research scientist in University of Cincinnati College of Medicine. He has done his undergraduate and postgraduate degree in botany from Vishwabharati. He is a PhD from Bose Institute, India, on allergy, aerobiology, biochemistry, molecular biology, immunology. He has a graduate certificate in biomedical informatics from University of Cincinnati, USA, as a feather to his cap. He has keen interest on identification of plant and animal and fungal proteins causing allergy, asthma, and other immune disorders. He elucidated the novel allergens and found out the atomic structures by spectroscopic methods. He was involved in discovering calcium-dependent enhanced mitochondrial biogenesis of bronchial smooth muscle to be a critical pathological factor in asthma pathology. Data analytics of gene expression signatures related to complex diseases have also been used by him. He is currently involved in multiple drug trials to better understand the molecular mechanism of diseases and drug actions. He is enjoined in instructing biomedical science students. He is the thesis examiner of Indian universities and is a mentor for European Academy of Allergy, Asthma, and Immunology. He has several patent applications and he has been awarded several times. He was awarded with GAT Fellowship from German Academic Exchange in 2002. He was also awarded the postdoctoral fellowship from the School of Medicine, France from 2002 to 2008, and he had been a scientist in INSERM Bordeaux, France from 2006 to 2007. He has also been a research associate in the same university from 2007 to 2011, and he is presently working as a research scientist in the University of Cincinnati in USA. He has several publications in international and national journals with very high impact values. He has experience in dealing with critically respiratory distressed COVID patients and have tried his drugs in a different way. So over to Dr. Devojyoti Ghosh.
So can I present the screen now?
Yeah, Dr. Ghosh, carry on with your presentations.
Okay, thank you. Can you see my first slide?
Dr. Ghosh, you have to present your screen. Share your screen.
Okay.
Yes, it's visible
Okay,
now.
good. Okay, thank you. So thanks for the kind introduction and good evening to everyone in India. The title of my presentation is, as you know, COVID-19, Myth, Science, and Hope. So I work as Dr. Maiti just introduced. I work in the University of Cincinnati College of Medicine, which is a major innovation hub for several biomedical products, including the polio vaccine that is administered now worldwide. So this statue is has been placed in the University of Cincinnati. It shows Dr. Albert Sabin, the inventor of the polio vaccine, the attenuated Sabin vaccine we are using now. He's administering to several children to combat polio. So in summary, I will present some of the myths today. What do you think, if they are true or false? The virus causing COVID-19 is a lab-made virus, is a man-made virus. Hydroxychloroquine is a wonder drug to fight COVID-19. PCR test and antibody tests are available for COVID-19. However, PCR is better. I don't have symptoms. I don't need social distancing. I am young and healthy, so I don't care. So, and there are a couple of other things about social distancing and our everyday life that people usually keep on asking me. Spraying disinfectants on sidewalks or on people like migrant workers can help stopping COVID-19. Wash vegetables with detergents. We need a proper N95 mask and a good branded hand sanitizer to stop infection. I received a packet from online shopping. Do I need to disinfect it? And finally, I have the virus in my refrigerator. So we will, down the line, during my presentation, we'll see what the research data says about it, about all these questions. So now COVID-19 science. So we will ask several questions during the my presentation, and we will try to find the answers. So in science, I have split into several subsections like epidemiology. So the question is, what is the source and trajectory of the disease COVID-19? Pathophysiology. What happens when SARS-CoV-2, the virus causing COVID-19, attacks? Molecular biology. The question is how is the viral genome and transcriptome look like? Hello? Am I audible?
Yes.
Yes, you are audible.
Okay.
Audible.
Okay, thanks. Molecular biology, the viral genome and transcriptome look like. Clinical immunology, the question is, what makes the patient so sick? And finally, current reality and hope. Is COVID-19 under control now? If not, what can we do about it? Now, first, some general remarks. So corona, the word corona comes from Greek meaning crown, and COVID-19 is a shortened form or abbreviated form of coronavirus disease 19, since it emerged in 2019. So it was first described, coronavirus was isolated from chicken in 1937, and during the 2016, it was described as a causal pathogen of several human diseases causing fever, cold, and like small diseases, everyday diseases, not so severe. So human coronaviruses were first identified in mid-1960s, mostly causing common cold. The pathogen causing COVID-19 is SARS-CoV-2. It is also known as 2019 nCoV or novel coronavirus or SARS-nCoV-2. So here is a tale of first two patients. Patient one, both of them actually showed up at the front desk at the outdoor of Wuhan Hospital, China, on December or during December. So for the first patient was a woman, 49 years old. She had complaints of cough and fever. She showed up at the front desk on December 23rd, 2019 with cough, chest discomfort. And then she came back when her symptoms worsened. She came and she got admitted on 2019, December 27th. And her pneumonia was detected by CT scan. But he recovered and he was discharged from the hospital on January 16, 2020. She was a retailer of the local seafood wholesale market. And patient 2, the second patient, he was a man, 61 years old. Just note the age difference. Man, 61 years old. He had a complaint of fever and cough. She came to the outdoor on December. He came to the outdoor on December 20th, 2019, with respiratory discharge. His respiratory distress started on. He could not breathe. And he again came back to the hospital on December 27th, and he got admitted to the hospital. He was in indoor, and then his condition worsened over next 2 days, at which time he was put on mechanical ventilation, and he died on January 9, 2020. He had been a frequent visitor of the local seafood market. So you can see the chest X-rays of the second patient who actually passed away, who expired. So the second patient on day 2, when he was first admitted after day 2, and it's the day 11th before his death. So, and you can see it's full of peribronchial fluid. Is a marker of infection, highly infectious pneumonia. So the full of peribronchial. So the CT scan, the chest X-ray is hazy because the chest is full of fluid, peribronchial fluid. So he expired. So, and they collected the fluid and did microscopic studies in China. So first they cultured bronchial epithelial cells and put a drop of the peribronchial fluid, and you can see here the result of the microscopy. A is mock, meaning they put some medium without the peribronchial fluid, and it's fine. But when they put a drop of the peribronchial fluid with a marked with arrow, it shows cytopathic effect. That means the cells are disturbed, they're killed. So microscopy showed cytopathic effects in human airway epithelial cell cultures after inoculation with the virus. So second, then they did transmission electron microscopy, and you can see the arrowheads. Here the arrowheads indicate extracellular viral particles. These are the viral particles you can see on electron microscopy, because you need an electron microscope to see viruses. And arrows, these arrows indicated inclusion bodies formed by viral components, and you have triangles that are ciliated cells. So you have a lot of viruses around. So these are the results of electron microscopy, showing first report of electron microscopy showing viruses. So they collected the genetic material and did a sequencing, next generation sequencing, and they found that it belongs to the family of known coronaviruses, which so you have the Wuhan 2019 viruses in red, and it has a similarity, genetic similarity with SARS-CoV-2 or SARS-CoV-1, the first SARS-CoV, which caused the SARS outbreak in 2003, and then in 2015 outbreak, it's called MARS, Middle East Respiratory Syndrome, or MARS. So it is a member of that big family. That's what they concluded. So what is in that wholesale market? If you go to the wholesale market, you will find items on sale: dogs, dog meats on sale, bat soups on sale. You can find live snakes and pangolins and bats. So you can buy those things. These are the livestock on sale. So these are known reservoirs of coronaviruses causing human diseases. But usually diseases caused by these viruses are very mild, causing cold and cough. But sometimes they can cause outbreaks, like in 2003 MARS, 2003 SARS, and 2012 MARS. So these are the bats and mice are the main reservoirs where the viruses replicate, and through a secondary host they reach human population. So like you have cows and civet cat. So 2003 2003 SARS, SARS virus used a secondary host called, they are all zoonotic viruses. The secondary host is civet cat. It's a kind of wild cat used as meat in China, some locations of China. And then camel. For 2012, it was camel, or which was the secondary host of MERS virus. So it's known. This is a typical structure of the coronaviruses. They have spike glycoprotein, they have hemagglutinin acetylase glycoprotein on the surface, they have membrane glycoproteins, small envelope glycoproteins, and nucleocapsids, and a single positive strand of RNA. We will come more about it later on. So the local authority closed the market, local seafood market, supposedly the main hotspot where the COVID-19 virus spread from. So and to control the outbreak. So now there is a little timeline of COVID-19 pandemic. So around the first week, the 5th of December 2019, the first cases were identified or reported in Wuhan, China. During at the end, the last week of December, the COVID, there was a lot of COVID patients showing up in the front desk of several hospitals in Wuhan. So Chinese authorities report outbreak of severe pneumonia to World Health Organization. And then during the first week of January 2020, so novel coronavirus identified in many patients, more patients causing pneumonia, and it spread in Italy and other countries of European Union through visitors or travelers. And then in the first week, in the last week of January, exactly on the 30th January, who declared public health emergency. And then on 21st of January of this year, the current year, the first case in the US was reported. So it reached US through a lady who traveled, who made a lot of travels in China, and she had relatives in China. So she arrived at the Washington State, United States, with the virus, and then the virus spreads. This is the first case in US, reported on 21st January this year. And 31st January, who declared global health emergencies because it already spread across the globe. And during like mid of February, World Health Organization named the new disease as coronavirus disease 19 or COVID-19. And then during the mid of March, a national emergency was declared in the US. So that's a brief timeline. And finally, COVID-19 has spread, as we all know, worldwide through travel and trade connections. So the first known severe illness caused by coronavirus emerged in 2003. It's known as severe acute respiratory syndrome, or SARS, and it was also spread from China. The second outbreak was reported as MERS and started in 2012 in Saudi Arabia, and known as MERS, as you all know, Middle East Respiratory Syndrome. So COVID-19 coronavirus epidemic probably has a natural origin, according to the scientific community here who are working on it. So SARS-CoV-2 is a lab-made or man-made virus. No proof, no direct proof yet. However, some people think that it can leak from a virus, from a lab, local lab who are engaged in research on these viruses, but there is no proof yet. So little bit of pathophysiology of the disease. So these are the symptoms and comorbidities we collected from B cohorts of COVID-19 patients. So when a people is exposed or infected, it takes 2 to 14 days, that's 2 weeks, to develop symptoms. The median time from infection to symptoms is 6 days. 5 to 6 days. So the major symptoms are fever, cough, shortness of breath, nausea, vomiting, diarrhea, taste and olfactory, that's smell and taste dysfunction. So vulnerable populations are people with cardiac disease, diabetes, pulmonary disease, and kidney disease. So age 65 plus is more often hospitalized. People have symptoms, mild to moderate symptoms, but mainly 65 plus people are more frequently hospitalized. However, younger people do get infected, get sick, and can carry viruses. So as you notice that the symptoms actually affects from origin from multiple organs. So because the virus which binds to virus actually uses a receptor called ACE2 protein, which is actually expressed on multiple human tissues, and SARS-CoV-2. That's why SARS-CoV-2 attacks multiple organs. You can see, if you look at this plot, you will see the expression of ACE2 receptor, which is a receptor for the virus, is expressed in multiple tissues like heart tissues, arteries, GI gastrointestinal tract tissues, small intestines, mainly smaller and upper lung airways, which is the entry point, major entry point of the viruses. So now the question is, did the patient, if a patient died, the question arises, the patient died from COVID-19 or died due to COVID-19 attack or died from comorbidities. So so to address these questions and to better understand the pathobiology, scientists or pathologists around the world has performed several autopsies. So in United States, they performed the autopsies in our Arizona facility, which is equipped for very highly infectious diseases, to handle highly infectious disease, dead bodies or corpse of highly infectious diseases. The autopsy shows this is the bronchial mucosa histopathology. So histopathological slide. This is the facility. This is the histopathological slide. So mucosa, it shows that mucosa is full of inflammatory cells, blood thrombus or clots, big clots in all organs that damages kidney, cardiovascular system, liver, lungs. So the patient is obviously died of COVID-19. But autopsy also said that it can damage multiple organs, not causing death. So autopsy can distinguish between both situations, but both situations are observed. Comorbidities actually can make this disease more severe. So this is an inflamed bronchial epithelium of a patient died of severe disease. So this is a summary slides of autopsy findings. You can see lung thrombosis and necrosis. So is a lot of blood clots everywhere. So spontaneous bleeding from arteries and veins, and systemic hematophagocytosis, a lot of phagocytes. Arterial and venous thrombi or clots everywhere. Clots in the lung arteries and lung tissues. Hemorrhages. So the arteries are ruptured, blood come out, make a lot of hemorrhages, and in organ, it causes a lot of organ damage. So pro-inflammatory cytokines and pro-coagulant factors. So blood clots, arteries ruptures, cells attack the blood cells, inflammatory cells attacks the host cells. So, and the cause of kills a lot of host cells, the infected cells. They make a lot of debris and in the lungs, and they cause they just disrupt the normal oxygen carbon dioxide gaseous exchange. So that's it leads to a symptoms. It leads to a lot of symptoms. The most severe is ARDS, acute respiratory distress syndromes. We'll come into that during the next few slides. So a little bit of molecular biology. So it's a short. The virus has a relatively small genome of 29.9 kb. So it has. This is the end term, the 5' end and 3' end. It has open reading frame 1 and 1A and 1B, which is followed by a lot of small protein coding genes. So these ORF 1A and B, open reading frame 1A and B, codes for about 16 non-structural proteins. So proteins are like non-structural proteins followed by S protein or spike proteins, and you see the spikes in famous spikes on the surface of the coronavirus. But all coronaviruses have the spikes. So in spike protein, envelope protein, protein M, protein N, and 3A, 3B, 6, 7A, 7B, 8, 9B, 9C, and 10. So these are the major proteins coded by the genome of coronavirus. So we'll come a little bit in a more about it. So next. So the coronavirus proteins, the viral proteins have known and unknown functions. So for example, non-structural protein NSP1, it slows down host protein production. NSP4 helps in viral protein assembly, so required for proliferation inside host cells. And viral proteases, they also have specific functions for viral proliferation. Protects NSP16 protects viral RNA from host degradation, because host has also RNAs which can cut the viral RNAs, degrade the viral RNA, kill the virus, and this protein protects viral RNAs. And then the major important protein for us, for researchers and everybody, is spike protein. So this is the structure of spike protein, which is involved in entry into the host cells. So, and there are a lot, the other proteins, the functions are partially known. So when the virus spreads across the globe, and it makes several mutations. So basically, this is the tree shows from the complete or near complete genome sequences showed several clades of the virus spreading across several countries. So the rate of mutations is about 25 mutations per year. So the genome is more stable compared to the normal flu, seasonal flu viral genome, which makes about 50 mutations per year. So you need the vaccines in each season actually. But since this genome is relatively more stable, we expect once the vaccine is developed, it will be effective for for longer time, for more viral strains. So this is the access point of the virus. The ACE2, as you all know, ACE2 is the receptor for the virus. This receptor is located on several tissues of human organs. So SARS-CoV-2 infects people by binding to ACE2 or angiotensin converting enzyme 2 using its spike proteins. ACE2 is expressed on epithelial cells in several organs, including the airways. Transmembrane peptide serine 2 or temprase 2 is a protease which partially cleaves the spike protein and primes it for binding. So it's another candidate is another protease is purine. They helps the entry of the virus into the cell. So this is a crystal structure showing the binding of ACE2, which is in green. The green ribbon is ACE2, and this purple is the spike protein protein residues, making a lot of contacts and making a lot of bonds and contacts. This is in comparison of the SARS-CoV, which caused a pandemic in 2003, the SARS pandemic. So it has been noted that the binding is more stable for SARS-CoV-2 or the novel coronavirus, because that which actually explains why it's more infectious than the previous SARS virus. So these are electron micrographs, transmission electron micrographs again, performed, obtained in CDC and in Singapore. So these blue dots are intracellular viral particles, and these circular dots are viruses attached to epithelial airway epithelial cells. So this is a little bit outline of how the virus proliferates in human cells. The virus is attached and engulfed in the goes into the human cells, and viral, it's a positive strand or plus strand, so it's directly used by the viral polymerases, RNA dependent RNA polymerases, to make the second strand or minus strand. So the RNA is replicated, and then it's transcribed and translated. It's actually not transcribed, translated into proteins, which all of these viral proteins are assembled in little vesicles inside the cells and released outside in the medium or in human body. So the matured viruses are released, but there are some proteins, like spike protein, which is highly antigenic, which is cut, which is cleaved by the host machinery and binds to MHC, or major histocompatibility factor of human cells, and present to other like T cells and B cells stimulations, and for creating antigenicity of the viruses. So two ways: the first, the virus is proliferated, and the second, some of the viral proteins, newly transcribed proteins, are newly translated proteins like S protein or N protein or nucleocapsid proteins, and the spike proteins. They are mainly antigenic. They stimulate, they actually elicit host immune reactions. So a little bit of clinical immunology now. So this is a common time frame of the viral immune response in human. So as you can see, that once the virus is actually entered the host affect the host, so it takes about 2 weeks so to get the symptoms. So detection, in the first, there are 2 weeks, so this is onset of symptoms. After the symptom starts, only after the symptom starts, there are a lot of viral particles in the blood or in the saliva or nasobronchial fluid, which is detected by PCR. So before that, the viral titer is low, so PCR detection is unlikely. You cannot detect by PCR. So once the symptom starts, at that time is the viral titer is high and detectable by PCR. So so it takes about like 1 week from onset of symptoms when to get the high viral titer in order to get it detectable by PCR. So, and still it is it takes more time to get an antibody response. So antibody detection is negative at that time. So after about 2 to 3 weeks, the antibody titer goes up. So the antibodies are usually IgM and IgG. So after 2 to 3 weeks, the antibody titer is high, and it is antibody detection is possible that time. But however, the viral titer that time goes down. So when the viral titer goes down, and usually it at some point about like 4 to 5 weeks of onset of symptoms, the viral titer is very low and again becomes undetectable by PCR. So however, there is the antibody response which lasts for like 6-7 weeks. So, and IgM goes down, and IgG titer, there is IgG antiviral antibody in human blood, which is about like three months after onset of symptoms. But more data is required in order to get any conclusive result. So however, once the person is infected, the antibody lasts for about 3 months. So now here it is. It's a typical pattern of the COVID-19 patients. So the first, viral PCR detection and the maximal infectivity. The virus, when the viral titer goes up, you can detect it by PCR tests, and there's a lot of viruses. Person goes on shedding the viruses, and at that time, a person is very a major source of infection. So, and then the viral titer goes down, PCR declines, PCR value, quantity PCR value, CT values declines, and viral PCR intermittent actually, and infectivity is low. Full recovery, if the patient recovers, full recovery takes about a month after disease onset. So mean duration from symptoms onset to death, and if the patient is it dies, it takes death typically occurs after 18 days after onset of symptoms. So after like 7 to 10 days, some patients, usually patients with comorbidities and older patients, they respond, their immune system becomes hyperactive. They moves on to an inflammatory phase, and usually death occurs like about 18 days of onset of symptoms. So mean duration from symptoms onset to death is 17.8 days, or to recovery and discharge is 24.7 days. So about 25 days. It takes 25 days to get fully recovered. And if the patient dies, death occurs about 18 days, in 18 days of onset of symptoms. This is the time frame. So I made a slide of like with a synopsis of the hallmarks of COVID-19. First is lymphopenia. Lymphocytes in the blood, lymphocyte blood count goes down because lymphocytes are usually recruited in tissues, causing inflammatory response, tissue damage. And then cytokine storms. That means all these blood inflammatory cells, particularly lymphocytes, monocytes, and macrophages, they produce a lot of huge amount of cytokines known as cytokine storm, and it also leads to an event called complement activation. So we have a complement immune system which gets also hyperactive, and which it particularly involves so-called, in immunology we call it contact pathway, which leads to clotting, blood clotting, and venous and arterial ruptures. So blood comes out, makes a lot of clots and cell debris. So which is technically known as thrombotic microangiopathy. So all the angio means the veins and arteries. So they ruptures and produce a lot of clots. And finally, ARDS, acute respiratory distress syndrome, which means that the alveoli are unable to make gaseous exchange, and that time artificial ventilation is required. All this leads to multi-organ failure potentially. So as you see, this is the blood test report. Interleukin-2 is very high in ICU patients compared to non-ICU patients, but COVID positive non-ICU patients and healthy. So mainly in cytokine storm, it's interleukin-2, macrophage inflammatory protein or MIP-1A, TNF alpha, interferon gamma, and so this is the hallmarks of cytokine storm. And the hallmark of lymphopenia is CD3, low CD3 count. So CD3 cells or a subset of lymphocyte goes down, and there is typically there is no change in B cells as well. So now I move on to reality. So as of July 26 today, confirmed death around the globe is 16 million, recovered 9.23 million, and death is 644,000. So new death in one day around the globe is about like more than 9,000. So it's really a matter to be worried. So now case fatality rate is really high. So these are the mainly in European countries, it has been very high all through. like, case fatality rate is the ratio between confirmed death and confirmed cases. So how many are the confirmed cases, and out of the confirmed, how many, what is the percent of death? So which has been all-time high in European countries like Italy, France, UK, and now the new candidates are Brazil, United States, India, Russia. So of course, this is not, there is a lot of factors behind it, like population median age. If the population, median age of the population is high, they are more prone to severe COVID-19 and potentially death. And genetics, immunity, environment, number of tests, sample size, small versus large. If you test a lot of people, this can change the number. And data recording, detection issues, health, and test population bias, because in some countries they're testing a general population. In some countries they are only testing the symptomatic people, like in United Kingdom they are testing a general population, everybody, trying to include everybody. But in other countries, if you are symptomatic, you can go ahead for testing. So there's a lot of test population bias. So so we can really conclude 100% on this. So, and daily confirmed death is high. Number of cumulative confirmed deaths is high in Brazil, United States, India, followed by these countries. So, so now, not everything, not all are bad news. There are some good news about medicine. Like, there are some potential candidates by remdesivir, So, like hydroxychloroquines and others, we will come into this, and then interleukin-6 inhibitors, and some others like interferon beta, and steroids recently made a lot of headlines. So, and finally, convalescent plasma. So people who are ill but recovered serves as a potential plasma donors, and supposedly they contain a lot of antibodies which can neutralize the virus. So convalescent plasma program has been developed in with collaboration industry, between industry and university collaboration. So 40 partners, they collect a lot of serum samples from this phase when they have a lot of antibodies, and they store those, trying to use in severe patients, and some hopeful results, but not uniform. We still need data on this. So now this is remdesivir is a analog which interferes with adenosine nucleoside triphosphate. So, and it's an antiviral. So we still don't have enough data to conclude on this. There are mixed results. So like hydroxychloroquine also made a lot of headlines nowadays. So historically it is used as an anti-malarial. So it suppresses pro-inflammatory cytokines, exerts direct antiviral effects, inhibits pH-dependent steps of viral proliferation inside host cells. Efficacy is, against COVID-19 is still debated. So previous studies showed hydroxychloroquine was effective in combination with azithromycin, but more recent studies in a randomized trial, it did not prevent illness. So the results reported, so there is one editorial published on all these results, and it says that the results are more provocative than definitive. So there is no definitive evidence that hydroxychloroquine can stop the COVID-19 related death or illness. So we don't recommend this at this point, usually at the clinics. So there are a lot of vaccine candidates are around, and these are usually developing vaccine takes about 2 years, but since several labs has experience on developing vaccine against SARS, the first SARS attack, SARS and MERS, so they're using the same technique, and some people are using repurposed drugs and vaccine candidates to make. Usually these vaccines are targeted against the spike protein or S protein, which is the main antigenic protein. So the only one mRNA vaccine is being developed in United States. The Oxford, another one is Oxford. In United States, it's called mRNA 1273. It's it's an stabilized mRNA again that codes for the prefusion form of S protein, which is absorbed in nanoparticles. It is being developed in United States. The other very potential candidate is the Oxford vaccine, which is also targeted against the S protein, but not much known about all these vaccines. Still need some more work on this. So now the main question is how to reduce ICU stay and avoid death. It's a very important area of research, and our lab is involved in this type of research. We are using recombinant complement immune system inhibitor, because a very important critical feature of COVID-19 disease is hyperactive immune system, particularly complement system, which activates the blood clotting pathways. So we are using several drugs to control to tame down or control the hyperactive immune system. So recombinant complement immune inhibitors, which actually stops the cleaving of factor 5A, which is famously known as membrane attack complex, because when it forms, the inflammatory cells attacks the host cells, and this is the major key point of COVID-19. So we are using some chemicals, synthetic peptides, to control this major step. So originally, these chemicals or the peptides were discovered from parasites who use these molecules to feed without alerting the host and provoking immune responses. So these are very useful to control hyperactive immune systems, so that the immune system does not attack the host organs and prevent. So these peptides actually can prevent organ damage. So another important candidate is dexamethasone, which is a steroid, which also can reduce inflammation and thereby reduce the risk of death. So finally, there are reasons of hope now. So prevention is better than cure, as we know. So case isolation and contact tracing, social distancing, these are the major steps of prevention: case isolation, contact tracing, social distancing, like keep 6 foot distance, 6 feet distance, limiting public interactions and gathering, maintain hand hygiene, avoid droplet contaminations. Because large droplets more than 100 micrometer radius it cannot fly long distance. So major droplets, medium droplets, 5 to 100 micrometer of 5 to 100 micrometer radius is the major cause of spreading of this virus, and small droplets at partially, so which has radius less than 5 microns. So they are mainly responsible for spreading. And do I need a mask, N95 masks or other masks? So there are some studies. It shows that there is no actual difference between N95 versus medical mask if you wear those masks properly. So it's the same as alcohol-based formulation for viral spreading. Usually, like more than 75% alcohol-based formulations can effectively kill the viruses quickly. So the question is then the refrigerator. What about the like vegetables and other things in the refrigerator? So there are some studies back with some kind of coronaviruses, which is not the novel coronaviruses. So it shows that if the sources of coronavirus are in the refrigerator, they can be alive or destroyed up to 30 days. But since vegetables are not major sources of coronaviruses, the chances of infection from refrigerator is minimal. However, with a note that if you have a lot of coronaviruses in the virus, they can stay good about 30 days, until about 30 days. And what about the packages from like online shopping or so? So these are not major sources. Again, not much to worry. It has been showed by several studies that the viruses on plastic or like papers, cardboards, can stay good maximum for like about 3 days, and then the viruses are denatured. So if you can leave those virus so packets or so for some time, they are safe to open. You can use a mask though. So now there are a lot of spread models. So like person A, when he is infected, he has no symptoms. That's very dangerous. He can, if he is not traced, he can can serve as a source of infection for example person B and person C, and then he can get isolated. So if he, and then the person B, when he has the virus, if he is traced, he can be isolated and the infection can be restricted. But if, for example, for person C who has the virus, who is not traced, we don't know, who is not tested, so he can spread the virus. All this event can be mathematically modeled. So, and showing that the contact tracing and testing is very important to stop the spread of the virus. Similar thing nowadays is making headline is herd immunity. So when a virus or a pathogen enters the population, there is no immunity first because it's novel. So it can spread very easily. There is no immunity. And there are some, at some point, some people are resistant, so they can effectively restrict the spread of viruses. So it's a mix. Some people are resistant, some are susceptible. But herd immunity comes when at least like 70 to 80% of people are immune, so it can break the chain of infection, so leading to herd immunity. Herd immunity has been seen in mumps. Their mumps and this type of diseases are now sporadic occurrence. They don't spread so much people because like 80% of people are resistant to mumps, so it does not spread. So this model did not hold good for these novel coronaviruses because they are so infectious and novel. It can kill a lot of people with severe disease. So this model failed in Sweden and in other countries, and not likely to work in India as well. So so that's what I have summarized in here. Chain of infection can stop when person is naturally immune or due to vaccination. Can at least 70 to 80% people become immune, condition known as herd immunity, and it happened in mumps polio. For COVID-19, it did not work in Sweden. So now some mythbusters. The virus causing COVID-19 is a lab-made virus, no proof. Hydroxychloroquine is a wonder drug, no proof. PCR test or antibody test, PCR is better, it depends. So when the person is infected, it takes like about 2 weeks, 1 to 2 weeks to get high viral titer in order to make it detectable by PCR. And then the viral titer falls down, antibody tests is then effective. So it depends. So I don't have symptoms. I can't. I don't need social distancing. It's false because you can still be a carrier. I'm young and healthy. I don't care. It's a dangerous attitude. So you need to wear mask, use hand sanitizers, keep social distancing. So spreading disinfectants on sidewalks, on people, washing vegetables with detergents, not much helpful. Maintain personal hygiene instead. So only a proper N95 mask and a good branded hand sanitizer can stop infection. False. Other masks and sanitizer can be helpful. So so we have reason to be helpful. Globally, about 3.4% of reported COVID-19 cases have died only. So more than 90% recovered. We know how to stop the spread. We know genome sequence of the pathogen and drugs to control, at least control the inflammation. Vaccine is on the way. Research is in progress at a lightning speed. So in the meantime, I would request everybody that everybody do your part. Don't spread infection. Don't get infected. Wear your mask properly. Keep social distancing. Use hand sanitizer and sanitize your hand. So what COVID-19 taught us is we made good progress, but still miles to go. We must understand our pathogens better. We must understand our immune system better. we must stop the spread. COVID-19 taught us that the show must go on. So as Louis Pasteur said, one must work, one must work. I have done what I could. And I thank you for your patient hearing. Thanks.
Thank you, sir. Thank you, Dr. Ghosh, for this well-programmed and well-discussed session. You discussed about the virus, which is under rigorous research. Thank you for discussing the difference between SARS and SARS-CoV-2. Thank you, sir, for discussing the spike proteins, the features of the coronavirus, their mutating nature, the idea about the receptor protein ACE2 and furin, which were found to be present in several organs and are the major entry points. You also discussed about comorbidity, the chances of attack, and also discussed about the clinical immunology. You discussed about the viral titer, the viral load, about the lasting of the antibody, and about the cytokine storm. Lastly, you discussed that there is hope because of the vaccines which are on their way and because of the medicines which are on their way. Lastly, you discussed about the myths of corona, and also you said that there is a chance of recovery, chance of getting well from corona, even when you practice the sanitization process.
Yes.
Thank you. Thank you, sir. Thank you, sir, for dealing with this tough subject in such a lucid manner.
Welcome.
Over to Dr. Ritu Puraparna Kundu Chaudhuri for the questionnaire session.
Thank you, Dr. Maiki. Indeed, it was a great pleasure to listen to Dr. Ghosh. You have explained in an excellent manner the structural, epidemiological, molecular, immunological, and hello. Okay. Hello. Is it okay?
No,
Is it okay now?
no.
Rituparna, the noise is still on.
Hello.
ফোনটা বন্ধ করো।
Hello. Hello. Is it okay? Hello. Is it okay now? Am I audible?
No.
I think it is because of the fan, probably.
But no, there is no sound from. Now
Right
is it
now
okay?
it's okay. Yes,
Okay,
yes.
okay. Okay. Thank you, Dr. Maiti. It was indeed a great pleasure to listen to Dr. Ghosh. You have explained in an excellent manner the structural, epidemiological, molecular, and immunological, as well as pathophysiological aspect of the virus, especially in relation to spike protein that interacts with human receptor ACE2 in a very elaborative manner. Thank you again for your candid talk. Now we shall move on to the question answer session. The few of questions are already awaiting our chat box, although many of our queries have already been answered by this discussion. Still we have a few. Number one question is, why the antibody, especially immunoglobulin G, is less in asymptomatic COVID-19 patients? Am I audible?
Yes.
Yes. May I repeat the question? Hello,
Dr. Ghosh.
Dr. Ghosh, may I repeat the question?
Dr. Ghosh, please unmute.
Hello.
Dr. Ghosh, please unmute.
Should I?
Yes, I can. Yeah, somebody.
Yeah.
Okay. So should I repeat the question?
I guess the question is why in some people, like in asymptomatic individuals, the IgG level is low. Is that your
Yes.
question? Yes.
Yeah.
Yeah, the answer is there is a lot of studies. In one sentence, I can summarize that the immune system in those individuals are not hyperreactive. In other words, they have a tolerance against COVID-19 antigenic proteins. So there's the searches are going on to understand why the immune system is not hyperactive. It can deal with a low level of sustained immunity. They are not
Okay.
hyperactive. So we don't understand this a lot, but it's known that their immune system in asymptomatic individual, the immune system is not very active against, like not hyperactive. It's the low level sustained immunity. A paper has been published on this in Nature Medicine early this year from Chinese groups.
Okay. Thank you, sir. The next question is, nCoV is continuously mutating and produces new strains. So how effectively the vaccine will be targeted against such infection?
Yeah, yes, as I have shown in one of my slides, that nCoV is genome is mutating as it spreads across countries and continents. But it's a RNA virus, and it depends on RNA-dependent RNA polymerase, so which has a proofreading activity. So it's unusual for this type of viruses. So the genome is more stable compared to other RNA viruses like flu, influenza virus. So it has a mutation rate 25 bases per year, which is compared to flu virus, which is like double, 50 mutations per year. So it's more stable. So for influenza virus, we have to make vaccines for each season for new strains. But for nCoV, it's more expected to once vaccinated, the immunity expected to last longer than flu. So actually, these type of mutant strains are usually not properly not called strains according to a strict sense. They are just isolates or clades, not strains. They are just mutant. So there are debates. It's not completely so different at this point.
Okay, thank you very much. The next question is, if antibodies remain for 3 months, then is there any chance of second time occurrence of COVID-19 during this period?
Yes, it has happened in Italy, in China, and I heard, and I have seen another report this morning. There is a chance.
Yes. Okay. And the next question is, interleukins mentioned also play an important role in cognition of human being. Whether COVID-19 affect the cognition of the affected person?
So I'm not very clear about the question. What is cognition? I mean the cognitive movements and recognition and this type of things, you said?
Role in cognition of human being. It has been presented in the chat box. I'm just presenting it to you.
The question is not very clear to me, but I can still address it. The problem is hyperactive immune system is a hallmark of this disease. So once the cells are infected, there's a cytokine storm, meaning that all these cells release huge amount of interleukins or whatever in the blood, and which disrupts all these contact pathways, clotting, and killing a lot of cells and making organ damage. Debris are gathered in lungs, in alveoli, disturbing the gaseous exchange in alveoli, and actually it has it can also generate cognitive problems. Yes.
Okay, thank you. The last question, as we are running very much short of time, we have to take our program again. We have to short the questions. So another question is, as you told about hallmark of COVID-19, apart from D-dimer, is there any biomarker can be possible to find out from complete blood count report of a susceptible person who is still not confirmed by RT-PCR positive report? If
No.
possible,
Yes, go ahead.
if possible, then it would be used as screening for isolation and quantitative susceptible, quarantine susceptible.
The first test would be PCR, like I said, because when a patient is infected, he does not have enough viral count to be detected. The first the first detectable thing is high viral titer. So when he has, and unfortunately it takes some time, about more than 2 weeks, to get that viral counter. The first marker is PCR, and then antibodies, and unfortunately the blood count, as you mentioned, it develops low, it's called lymphopenia. So lymphocyte count goes down. So it actually develops late, and that by that time it can be potentially very late for the patient to use is as an early marker. It's a late marker of the disease. So first should be PCR, then antibody, and the serologic marker is D-dimer, but it's for high risk severe patients and lymphopenia.
Okay, thank you very much. And last question for you is, what is your comment about favipiravir? How it would affect to reduce the viral load?
Oh, well, we don't work with this drug, but initially it gave some. It is actually a repurposed drug, so it was not used before. There are some trials going on to use it for this novel coronavirus, but we don't have enough data to make any definitive conclusion about it. It's a repurposed drug. It was first developed but was not used. Recently it's repurposed for SARS-CoV-2, but we don't have real data about how it's doing.
Okay, so thank you very much, sir. I'm very much grateful to you for sharing your knowledge and valuable time with us. Surely our students and all the participants will be immensely benefited from this. And with this, I would like to conclude this question and answer session. And now I would request Dr. Maiti to introduce our next speaker, Dr. Monishankar Bhattacharya.
And thank you, Dr. Ghosh. Now, the second speaker, Dr. Monishankar Bhattacharya, has already joined us. May I please introduce Manishankar Bhattacharya. Dr. Manishankar Bhattacharya, Senior Scientist in Institute of Microbial Technology, a CSIR institute. He joined as a scientist in the same institute in 2011. He did his undergraduate and postgraduate studies in Vishwabharati and submitted his thesis in National Institute of Pharmaceutical Education and Research, NIPER, Mohali, in the Department of Pharmaceutical Technology and Biotechnology. He has been awarded with several national scholarships and fellowships during his graduate and postgraduate studies from the Government of India. He remained as a MRUR Fellow from 2006 to 2009 from Italian Ministry of University and Research. He also had a training in advanced course in downstream processing from Technological University Netherlands in 2007. As an assistant professor of Academy of Scientific and Innovative Research, Chandigarh, and IMTech JNU joint venture PhD program, he also teaches basic organic chemistry, bioinstrumentation and biotechniques, bioorganic chemistry, and biochemical engineering. His research interests are fermentation of surface tension lowering and micelles forming compounds, and downstream processing, biocatalysis, immobilization, sterilization, development of novel antifungal compounds, development of antifungal and antibiofilm products, application of nanostructured materials in biocatalysis and drug delivery. Two of his PhD students have obtained their PhD degrees under his supervision. Presently, he is guiding 8 students as guide or co-guide. Manishankar is actively engaged in school student outreach program through Jigyasa, providing training to the masters and graduate students through internship program. I welcome you, sir. It is a great honor that you have accepted and given your consent in spite of your busy schedule. Over to Dr. Manishankar Bhattacharya. Dr. Bhattacharya.
Thank you, Dr. Maiti, for your kind words. And it's really a big introduction. Let me upload my presentation.
Yeah. We have to wait. His name is not there even in the list. Manas De, M-A-N, only one is there, man. Sharmishta, can you contact him over telephone?
Yes, I am contacting.
Okay. তোমার এখানে স্পিকারটা বন্ধ করে দাও, কথা বলে নাও দিয়ে তারপরে।
Okay, he is uploading his lecture.
Okay. That's why disconnected.
Yes.
So, Devajyoti, you did MSc from Botany department. Hello, Devajyoti, can you hear me? Devajyoti, can you hear me?
Okay. Yes, go ahead.
You are from Botany Department, Vishwabharati?
Yes.
From which year you did?
97 passed out.
I am from zoology. And I was in Cleveland. And into that told about you. At that time, I came.
Kaushik, Kaushik, Moni is facing some problems regarding sharing his files. So he is sending his files to you by mail. So can you upload or share those files? Okay, oh, he is sending you. এই স্পিকার এটা মাইক্রোফোনটাও অফ করা আছে দেখছি।
Mm. Yes, I'm unmuted. Audible?
Yes. Yes, Dr. Bhattacharya, now we can hear you.
Yeah. Hello. Am I audible?
Yes, you're audible.
Okay, so can I start?
Yes, yes.
So after a initial hiccup, we should start probably right now. So can you see the slides?
No, the slides are not visible yet.
Slides are not visible yet.
Hmm.
If you can stop presentation and again share your screen, I think it will be.
Yeah. Stop presentation. Oh.
On the lower right, you can see you are presenting.
Entire screen go. Is it visible?
No,
No,
not yet.
no,
Not yet.
So, in which medium your presentation material is? Is it in PowerPoint?
Yes, it is in PowerPoint.
So instead of presenting your entire screen, you present a window.
Windows.
Go into presentation in the top lower right corner, and you try presenting a window.
First, do stop sharing. The stop presenting is still.
No, sharing has not been stopped.
First stop sharing.
First stop sharing.
sharing.
Okay.
Then go at the lower right corner.
Is it visible?
No, it's still in the sharing.
No.
Okay.
Monishankar, I think you have a network issue. You can turn on your mobile and use the mobile network by your hotspot.
Bolo. Yes. I'm trying that mobile network.
Yeah, this is now absolutely fine. Now present now. There is an option at the bottom, present now. At the right bottom.
Let me check the right button. This is it. So, can you see the presentation right now?
No.
No presentation.
No. So you cross the meeting detail at the top right side. There is a meeting detail. You cancel it.
Meeting details. Edit.
Now
Top right side meeting details.
just there is a crossbar there. So cross it.
Yes.
Now you can see the present now at the bottom right.
Yes.
Click it.
Then Windows.
No. Yeah, you can do window. You can do entire whatever you want.
So let us go for.
It should be entire screen. It would be better actually, because
Let's go to the entire screen.
Because nothing is there now with other than PowerPoint in your computer is open.
And go to the entire screen.
Go to the entire screen. Your entire screen.
Oh my God.
Do you see the entire screen?
Yeah. But on the entire screen, nothing is coming. But
I think then go to the window.
Yeah, just a moment.
Yes, absolutely fine.
Got it.
Now click the that slide. Full mode. Full screen mode.
Full screen is in the top right, just beside share, yellow share button. There is a present button.
Top right.
Just beside there is a present button.
Is it a full screen right now?
No.
No, actually slide doesn't even move. No.
I think you can go with this thing.
Then it will be full screen.
No, I have already tried all these things.
Munishankar, you can carry on with this thing also. We can read. We can see.
It is possible to read, but I think it is not moving in that way. Whatever I want.
Yeah, that would be the only problem. But otherwise, you can explain.
You can hear that again? Okay. Let me try. No, magnify is it's not working. No. Okay. So anyway, let us start. Hello. Yeah, I think.
Okay, sir, you can start.
You can see these things. I can well manage this part because it's not working in that way. We are running out of time.
Yeah.
Yeah. So let me continue. So basically, I work in Institute of Microbial Technology, which is a new establishment, one of the newest establishment of CSIR. It was established in 1984 when there was insurgency problem in Punjab. So Ministry of Science and Technology that time thought to establish some institute in Punjab, and also there was a need for indigenous technologies based on microbial products like antibiotics and some other products that we still import from China. So that with that mandate, this institute was established.
Munishankar, you have to.
Hello. Yeah, please.
Please go ahead. It's fine.
Okay. So we you can listen it. No, you can hear it. Okay. So the thing is that while establishing this institute, certain mandates were fixed, and these mandates were like that. Since we are having some technologies maybe at a primitive stage, so we must optimize the existing microbial processes to make it available to the country. And for that, Biochemical Engineering Research and Process Development Center was established in that institute. Then to develop and maintain the gene pools and resources, we established microbial type culture collection, because that time only in NCL Pune there was a rudimentary center for microbial type culture collection. Later on, to carry out research on microbial products and cutting edge research on various aspects of microbiology, various centers were established, and among them instrumentation center was there. In late 2000, early 2000, it was felt that there must be a center for data curation and bioinformatics related center. So we established Bioinformatics Center, which is one of the most accessible data center in the world. After this is the second largest data access center of the world, and later on various other centers like Protein Research Center and Bio and Business Development Centers were established. So by training, though I have training on biology, but somehow I am related to the biochemical engineering and process research development. Let's come to the topic of today. So we have already gone through we have already heard Dr. Ghosh, and in that case, what we have heard is that how coronavirus infects and all this story. So let's just to go through the introductory part. Coronavirus obviously is having a corona over it, and that corona is for its name, is responsible for its name. So if we classify the virus, generally we have 6 human coronaviruses, and one group is subdivided into 2. So altogether we have 7 strains. Structure wise, we have with it some outside, that is corona protein, that is spike proteins. Then after that we have nuclear protein, we have envelope protein, and we have membrane protein. Every structure of that has definite function, and Dr. Ghosh has already explained these functions. So for other type of coronaviruses, probably the same slide has been also shared by Dr. Ghosh. What I would emphasize is that every cases there is almost out of that 7 cases, that 4 cases is having known intermediate host, and these are animals other than human being. So basically this coronavirus is a zoonotic virus. We always need a separate host apart from human being, and this is just the classification of the virus. Next we come to the topic of the actual topic of today's talk. So we are talking about the myths, sciences, and hopes of this corona pandemic or COVID pandemic. There are different kind of myths that are prevailing in the society, and there are myths that are also prevailing in the scientific community. So first thing is that we have heard that this for this kind of viruses, community spread may occur. Means, does it mean that wherever we go, it is we can get infected with corona? Obviously, it's not like that. The term community infection means that spread occurs, but the actual source of infection is untraced. However, transmission occurs knowingly or unknowingly through a contact person. We don't know that contact person, and community spread of infections can be ameliorated or can be stopped by using good practices like good hand hygiene, home quarantine, being at home, and also using hand sanitizer and other means of keeping ourselves away of these viruses. So the next thing is that whether COVID-19 is transmissible and deadlier than flu. The statistics says that the fatality of flu is less than 1%, whereas for SARS-CoV-2, the fatality is around 2% till date. So data demonstrate that SARS-CoV-2 is more transmissible, and it has higher infectivity compared to the common flu. Now, flu or pneumonia vaccine that we use can also help in preventing COVID. This is another question that we face, and there is a kind of social ideology or social thinking that probably it can help us from getting COVID infection. But the thing is that influenza is a different thing, and pneumococcal, the pneumonia that develops is due to pneumococcal infection. So the vaccine essentially is inefficient from protecting us from COVID-19. On the other hand, though the simple symptoms of COVID-19 are quite similar with influenza and streptococcal infection, they cannot contribute to the stopping of COVID-19 infection. So another myth that is prevailing is that probably summer comes and we will be getting relieved of corona. It doesn't happen, because basically the spreading of the disease occurs through contact infection. And also, if you think about our northern hemisphere and then southern hemisphere, then when in the northern hemisphere there is summer, in the southern hemisphere there is winter. On the other hand, if you think about MERS infection, that MERS pandemic occurred in 2012-13, that happened actually in very hot countries. So temperature has nothing to do with that. Another myth that is prevailing is that whether COVID-19 vaccine will end the risk of pandemic. Now, this is a very big question and it poses a big caveat of discussion. Now, some vaccines, it has been shown that some vaccines worsen the consequence of infection rather than protecting, a phenomena which is called as antibody dependent enhancement. So there is a report in Nature Medicine, which has been cited by Scientific American, that in 1916, a dengue virus indeed was given to protect the children from dengue, but it led to the hospitalization of the children who received the vaccination. Therefore, we should not rush for vaccination as we are doing right now. So, the sixth you can say is that asymptomatic not at risk. Obviously, they are at risk. According to Center for Disease Control, up to 20% of the between 20 to 44 end up in hospitalization because of the disease. But even people with asymptomatic infections are left with serious consequences and damage of the various tissue and organs from this disease. A study published in Nature Medicine shows that 2/3 of those with no clinical signs of COVID-19 had ground glass opacity in at least one lung. What is ground glass opacity? If you subject the people or person who is infected in that condition to radiological test, the CT says that there is opaque region within the lung which are either of liquid material or maybe there is some solid material, but they are non-functional. So the lung is damaged. With each and every statement, at the bottom I have given the references. So people who are not in the direct scientific field, they can have a photograph or can contact me offline to get these informations. So now the question is that how the coronavirus actually enters the cell and creates the problem. Dr. Ghosh's lecture has already illuminated us regarding these things. But still, I would like to give you some idea because this will help us later on in understanding how the COVID virus, how the drugs are being developed and how the virus is being targeted. So the entire process of entire life cycle understanding is very important. Now, these viruses have spike protein, and in our body there are receptors for different viruses, different coronaviruses. We have different kind of receptors. Like for SARS, we have angiotensin converting enzyme 2, that is SE2. On the other hand, MERS, we have dipeptyl peptidase 4, that is DPP4. So by binding the receptor, it enters the cell by phagocytosis or pinocytosis, and when it enters the cell, the viral proteins and capsids are destroyed by the proteases. Then the mRNA is released. When the mRNA is released, this is a positive sense. Sorry, not mRNA. This is the RNA of the virus is released. This is the positive sense RNA, and which is get translated. Therefore, the RNA-dependent RNA polymerase comes in picture, and the antisense RNA is synthesized. Thereafter, a host of proteins, that is nucleocapsid, membrane protein, envelope, spike protein. These all are synthesized. Then this protein goes to the ER, endoplasmic reticulum, then Golgi, and then from Golgi with the RNA again, it assembles, reassembles, and then breaks the cells and gets spread. But the caveat over here is that how the virus particles get transported to the other organs. Now, it has been proposed that there may be microsomes that actually carries them through blood to the various organs, and therefore we can see damage, hepatocytes, that is liver damage, pancreasocytes, and also kidney damage. But still, this is to be established experimentally. This is at least at the present stage at the theoretical understanding level. Let's go to the immunological part of this. I'm citing here a very recent article which has been published in Science on 15 July 2020 that analyzed 125 COVID patients and compared the recovered to healthy individuals using high dimensional cytometry. They have used a huge lot of cytometry. So the integrated analysis of around 200 immune and 50 clinical features revealed activation of T cells and B cell subsets in a proportion of percent. A subgroup of patients had T cells activation characteristic to acute viral infection and plasmablast responses reaching up to more than 30% of circulating B cells. Another subgroup had lymphocyte activation comparable to uninfected subjects. These immune subtypes probably in future may help in designing therapeutics and vaccine against COVID-19. So what we learned from that paper is that acute SARS-CoV infection actually results in broad change in circulating immune cells in the body. What happens? It is associated with CD positive, CD8 positive T cell activation in a subset of patients. On the other hand, in another subset, heterogeneous CD4 T cell responses occurs and activation of CD4 cell subsets also occurs. Therefore, this infection is associated with increased frequencies of plasmablasts and proliferation of memory B cells in the subset. Thus, a temporal change in the immune cell population occurs during the COVID-19 disease. That is actually leading to the immune cell, altered immune responses in the COVID infection. So, let us go to the therapeutic approaches. So, with that knowledge, what we learned is that there are several steps in the infection of the virus. So these infections are binding with the cells, then getting in, then replication, then the destruction of the outer layer, and then release of the RNA, then its transcription, replication and transcription, and then assembly. So various stages of this infectivity has been targeted, and we have got several molecules obviously repurposed for these things. Why repurposed? Because repurposing is being done because of the fact that to minimize the time for testing the drugs at various stages. So for viral attachment and entry, we have got chloroquine, then camostat and nefamostat. Then for viral proteolysis, we have lopinavir, retinavir, and disulfiram. So viral replication, to stop the viral replication, obviously we have remdesivir and favipiravir. Cyclosporin A basically an immunosuppressant which is also used, and for immune antibody treatment, also we have tocilizumab and sarilumab. Now, so let us in this slide, I can I wanted to just give you an idea regarding the targets, where various targets could be done, and then also what happens if the virus is not contaminated. I mean not contained, means if the replication of the virus is continuous. If the replication of virus is continuous in the host, then a lot of cytokine related changes occurs, and other activities also occurs, like activation of PPRS, then caspase 11, caspase 1, and TMEM things, and therefore we can see a cytokine surge, especially interleukin 6, and that actually leads to the other consequences of the disease that we know already. Now, regarding the hope, let's see, in India, we have already in news that there are various vaccines are coming, and the news is that almost 10 vaccines are coming in market. Out of that 10, at least 4 have originated from India. That is that information is available from the WHO landscape. So India is also not I mean behind in the run of vaccine against COVID pandemic. So I am associated with CSIR, so I must give some idea about the activities that CSIR is doing, and also it will be good for the people who are listening to me regarding the activities that we are doing within our country, because obviously throughout the world various countries are doing their research at various levels, and India is also not behind. So India is having a typical population landscape and also economic landscape. Therefore, the Council of Scientific and Industrial Research has approached with various institutes of its sister institutes to do various research, and at Institute of Microbial Technology we are basically involved with the testing of the virus, then sequencing of the virus, and also for testing of the newly developed molecule, because we have facilities over here for virus culture and we have already established it over here. So our establishment is showing that the virus that we are culturing over here is taking a peak of within around 21 days, and various drugs, drug candidates are being tested over here. As a whole, CSIR is providing a platform for technologies to the country, and we are working in various lines like digital and molecular surveillance, then rapid and economic diagnostics development, new and repurposed drug synthesis, and also vaccine generation, then developing hospital assistive devices and PPEs, and also to support the supply and logistic chains for mitigating the problem in the country. So just to name, for rapid and economic diagnostics, we have dry swab based RNA extraction kit, you know, for the testing of these things. The RNA the virus has to be neutralized, then RNA has to be extracted, and then the RT-PCR has to be done. It's a very lengthy process, so we have to have something which is very easy to do and which is not very lengthy process. Then we have a free direct RT-PCR diagnostics, that is Cas9 based diagnostics, which got populated into the media also, named Feluda, which has been developed by IGIB. Then we have IgG and IgM detection using a biolayer interferometry. Then micro-PCR based disposable chip, NGS based high throughput diagnostics, then PCR kit for COVID-19, and then also LAMP diagnostics of COVID-19. For development of repurposed drugs and new drugs, we are already having processes for the nitazoxanide and then chemostat also. Then we have synthetic processes for chemostat. This is basically a salt, a methylate salt. Then we have umifenovir, which is being tested, and also telurone dihydrochloride for all these things. So we have several drugs in pipeline that is being developed by the CSIR itself. Apart from that, CSIR is also collaborating with the other pharmaceutical giants in India for the development of other molecules, and also for the synthesis for providing assistance for the synthesis of some other drugs that are coming in the market. So to assist the hospital requirement and personal, we have developed various devices like ventilators, then aerosol canopy that is basically used for dental procedures. Then we have oxygen enrichment unit. We have positive airway pressure developing system. Then we have efficient antimicrobial materials for different kind of formulations, coatings, fabrications, and these things. Then we have modular and makeshift kind of hospitals and tents to attend the patients in various areas, remote areas especially. Then for hospital care, the robotics has been also developed, and then 3D printed various devices that are used in hospital that has been developed through 3D printing. So these technologies are already out. We have some other portfolios on which work is going on. Now, what we do in our lab? When I started my work over here in IMTech, I got a responsibility to do something new. I had a training on biocatalysis, I had a training on enzymology, I had a training on downstream processing, fermentation, and everything. So my director wanted me to work with something which is having medical significance, but in India, too much work is not done. So I found it is as a biochemical engineering part, I found it is I should take up some problem that is pertaining to for the biochemical engineering part. So I started thinking of something which is actually the fermentation of the molecule is actually giving problem, because in that case I chose a biosurfactant. Now biosurfactant actually changes the surface tension of the liquid, and therefore the mixing parameters and the nutritional parameters, then mass transfer parameters in biochemical engineering and fermentation changes. Therefore, when I choose superliquid, the reason behind that was this molecule had a huge biomedical application too, apart from being a surfactant. So recently we have found that this is a molecule which is having virucidal and spermicidal effect. So this can also improve the sepsis symptom, ameliorate the sepsis symptoms, and therefore it may function against COVID infection if we can develop the molecules by decorating it or by changing its structure. So we have already proven that this molecule could be used for various purposes, as antimicrobial, as antifungal through several publications. Right now what we are trying is that previously we developed a catalyst by engineering it and then immobilizing it on gold nanoparticle. Then we are trying to attach it with this molecule, so that this is not a single molecule, we have a repertoire of molecule, so that we can develop a number of compounds that can work against COVID, at least as antiviral, and that can stop the viruses. So what we have, the approach we have taken is that first we have done some molecular modeling with the help of some chemists of the institute, and then we are trying to design it using this biocatalytic processes. So next, I would like to give you some idea regarding the testing of COVID for the people who are not actually related with this kind of things, and basically laymen. For them, I just wanted to say that we do first, we receive the samples over here, then we actually carry out, we actually kill the virus by adding some reagents, and then extract the RNA of the virus. Then for screening, we do RNA gene, then an E gene as positive, and then once the screening test give positive for coronavirus, at least this gives the positive or negativity for class of coronavirus, then we go for the confirmatory test. In the confirmatory test, what we do, we test the RNA dependent presence of RNA dependent RNA polymerase, and then if it shows positivity, then we say the patient is positive with corona infection. So this slide summarizes the whole procedure. That is the screening. We have RNase gene as a RNase P, actually basically it's a positive control, and then we have E gene. If it comes positive, then we go for confirmatory test, and in confirmatory test we have we look for the RDPD gene, which gives us positivity or negativity. So finally, this is my group, and in my group presently I am having 4 PhD students. My first PhD student, Farazul Haq, who is right now in as a postdoc working in Japan. Now I am specially mentioning this because of the fact that Dr. Ghosh during his talk told that whether there was a debate or a myth that whether the virus has been originated from Wuhan lab or it's a virus that has been created or not. So with that, the name of a Nobel laureate Tosoko Honjo was attached, and this guy Faraj works with Dr. Tosoko Honjo. So when I contacted with Faraj, I asked him, what is your opinion? Why the name of Dr. Honjo is coming with this kind of things? He clearly confirmed me that Dr. Honjo never visited China and never worked in Wuhan. So this kind of things are prevailing in the internet. Please be aware of that. These are all myths. And then these are all PhD students like Nitish, then Sandaldeep, then Ankita and Yashna is working. So Srishti and Sachin has already submitted his thesis. Nitish is at his final year, and Jagdish has worked with me as a postdoctoral fellow. So he was one of the backbone in establishing my research in antifungal development, and that takes me to the end of this presentation. So I would like to end this presentation with a note that development of anti-COVID is possibly the single biggest challenge faced by the world in the area of infectious disease presently. So thank you very much. I would be happy to have the questions.
Thank you, Dr. Manishankar Bhattacharya, for this informative and enchanting talk of yours, which enlightened us on the science, on the myths, science, and the hopes of coronavirus. We discussed about the several diagnostic kits, the repurposed drugs, and also you discussed about the various technologies adopted by CSIR to combat corona. And over to Dr. Rituparna Kundu-Choudhury for the questionnaire session.
Thank you, Dr. Maiti. Thank you again, Dr. Bhattacharya, for your splendid explanations. Hopefully, all the listeners will be benefited from this. We have a number of few questions. So the first question is, how many clades or subtypes are present in India or globally of this coronavirus?
First, I would like to apologize from my part for getting this snag or this hiccup for the initiation of the presentation. The thing is that it's very preliminary. It will be very preliminary to speak about these subtypes and these things because still data are yet to come. But the thing is that whatever things we are getting right now, information we are getting right now from various parts of the corner is like that. In present time, there are at least 3 varieties that we are seeing. One is basically the subtypes which has originated in Yuan, that is known as Yuan type, and second one is basically the subtype which is prevailing in Europe, various parts of Europe, which is known as Europe type or Italian type or something like that. These two are actually causing more fatalities and morbidities throughout the population, especially in India. But still in India, another subtype is prevailing, which is known as Southeast Asia type, which is actually not that much fatal as claimed by the researchers, but still confirmatory information is yet to become. From the unconfirmed sources, I can speak that the virus is having different mutation at different level, but still we have found around 11 mutations, and that cannot be designated as different strains or different subtypes. That I can say.
Okay, thank you very much, sir. The next question is S2 receptor present in kidney and lung, but why lung is more prone to COVID-19?
Because it is exposed to that virus. You are aspirating it through the droplets, getting these things first be seated on your lining of the throat, and from there it can directly go to the airways, through the airways to your lung. But for the passage of kidney, it has to be transported by some mechanism, and that mechanism is yet to be revealed. People are talking about microsome mediated transport, but still that has to be established.
Okay. So thank you, sir, very much. With this, I would like to conclude our question and answer session at this position, at this moment. I would like to request our head of the department, Dr. Pintu Banerjee, to present the vote of thanks to both our speakers and all. Thank you very much. Over to Dr. Pintu Banerjee.
Good evening, everybody. I feel privileged to express vote of thanks for this international webinar entitled The COVID-19 Pandemic, Myths, Science and Hope. It's my great pleasure and opportunity to convey my sincere gratitude to the speakers who delivered their lecture on this topic and enlightened us about the matter. I want to extend thanks to the first speaker, Dr. Debojyoti Ghosh, Fellow, the American Academy of Allergy, Asthma, Immunology, University of Cincinnati College of Medicine, USA, who is very busy with his research work, has very nicely discussed the story of COVID-19 all around 360 degree, understandable to the audience from any discipline. He unveiled the myths of the enigmatic COVID-19 into reality in the light of modern biological science and shown us the avenues of hopes. My heartfelt thanks to another speaker, Dr. Moni Shankar Bhattacharya, Senior Scientist of CSIR Institute of Microbial Technology, Chandigarh. In spite of his busy schedule, he has consented to deliver a lecture and enriched our knowledge in this specific area. He very beautifully discussed the topic, very critically, taking the title, The Myths, the Science, and the Hopes. So, we do believe that the morning sun will glow on all the corners of the globe very soon. So thanks to Dr. Bhattacharya and Dr. Ghosh once again for their very nice presentation. Now, I convey my sincere gratitude and thanks to our Honorable OIC Sir and President of this webinar, Dr. Shobhan Neogi Sir. With his huge administrative workload during this lockdown period also, he constantly encouraged and supported us to organize such type of academic activities. I extend my heartiest thanks to the Vice President of this webinar, Dr. Amoresh Mondal, the IQAC coordinator, for his inspiration and support. I want to acknowledge our Chief Advisor, Dr. Kalidas Das, for his creative and constructive suggestions. I want to convey my thanks to the coordinator and joint coordinator, Dr. Somishta Maiti and Dr. Rituparna Kundu Chaudhuri, who are the main pillars of the Department of Botany. They always help me to discharge the departmental activities in every way as and when required. Dr. Maiti and Dr. Kundu Chaudhuri very nicely organized the program, and thanks to them. Dr. Sutoba Sannal, an IQAC member, has sincerely performed her role as convener, so a lot of thanks to her. I want to thank our departmental colleague, Dr. Sumanjana Khatua, an organizing member of this webinar, for her active participation, support, and cooperation. Now, I want to convey my heartfelt thanks, love, and affection to the Joint Secretary of this webinar, Dr. Kaushik Sarkar, without whose active support and constant effort, we were unable to organize this webinar. So I personally and on behalf of the Department of Botany as well as the organizing committee, I convey him thanks. I also extend thanks equally to Dr. Krishnendu Sarkar for technical support for this webinar. Thanks are due to all my colleagues, supporting staffs, and beloved students who are present here. Last but not the least, I extend my sincere gratitude and thanks to all the participants who are watching and listening this webinar since 6:30 p.m. in Indian time to make this webinar a grand success. Thank you once again. Thank you.
Thank you, sir. And the session is over here. Thank you.